All right, good morning, and welcome to part four in my biology series. The first of the ones that I have recorded from home. Good old remote learning, and as you can tell from the title, today we're going to be focusing on cell membrane structure and membrane transport. It's probably one of the bigger topics for Unit 1 biology, and it's definitely something that carries on into Year 12 biology as well. So it's important that we get the concepts correct. I'm going to go into a little bit more detail than the Year 11 program generally dictates, but hey, I have that flexibility because I'm making videos. I'm going to see how I do in doing this in one take as well, so that could be very entertaining. So let's get started. So, our last lesson, we looked at parts of the cell, and one of the things was the cell membrane. And why is it important that we have this cell membrane? Well, we know that it creates a barrier, but if we start to have a look at what's going on inside and outside of the cell, and if we're thinking about those words are going to be, if this is inside the cell, this is intracellular, and outside the cell is extracellular. Okay, then we can start to see why the balance is so important. Now, because I've written these almost a lot in the way that the animations work, I do have to get rid of the words. But yeah, if we start to have a bit of a look, well, we can see that, we look at some of the dissolved substances, or the molecules that exist in our bodies. Potassium exists inside the cell at around about that 120 millimoles per litre. Outside of the cell, it's only about 4.5 millimoles. Inside the cell, we're going to have about 15 millimoles of sodium. Outside, I think it's meant to be 150. Sorry, that's 150 millimoles outside. But yes, okay. First error. Whoops. Calcium, 10 millimoles per litre inside, about 1.2 outside, and chloride is at 20 millimoles per litre inside, and about 116 outside. So the first thing you have to notice is there is a difference, minus, of course, my error here that I've done. This is meant to be 150 millimoles, not 15. And that is that this is constantly changing. So there's things going across this barrier all the time. And it's important that we keep this balance. Keeping this equilibrium is one of the key factors of, or key roles of the plasma membrane. So if we zoom in on a bit of the plasma membrane, we're going to have a look at one of these funny looking characters. This is the one that I've done as for the animations, and they don't exactly look exactly like this, but hey, this is the type of structure that you will be finding as part of the plasma membrane. And this fella is known as a phospholipid. And it's the main structural unit of the plasma membrane. It's what gives the plasma membrane its function, its structure and function. It's made up of two parts. The first part is a phosphate group, and a lot of biologists and biology teachers will call this a phosphate head, and the phosphate head, because it's a phosphate group, is hydrophilic. If you remember back to our lesson on water, what hydrophilic and hydrophobic mean. Whoopsie. Hydrophilic and hydrophobic. Hydrophilic is water loving. So these guys love water. They are attracted to water. They are a polar molecule. And they are attracted to the water molecules. It's also made up of two fatty acid tails, or fatty acid groups. We call them fatty acid tails. So we have a phosphate head and fatty acid tails. So there's two of those in our phospholipid. These guys are hydrophobic. Forgetting how to spell for a minute there. All right, so hydrophobic being water hating. This is really, really important for the structure of the plasma membrane, because our plasma membranes don't exist as one singular phospholipid. They exist as many of them in what we call a bilayer. Do that again. So we end up with this phospholipid bilayer where we end up with two regions. We end up with this middle region that is non-polar, hydrophobic, and these two bordering regions, which are polar and hydrophilic. So we have these heads that are pointing outwards, this middle zone, which is non-polar, and again, this is going to be really, really important for the structure of how the plasma membrane functions. Now, the plasma membrane isn't just on its. The phospholipid bilayer isn't the only thing that's in the plasma membrane. In fact, it's a bit of a puzzle. Cameo appearance. There's a bit of a puzzle going on in the background here. What we're going to end up with is... So the phospholipid bilayer doesn't just exist on its own. It is in fact part of a mosaic. It's part of many different parts all into that make up the plasma membrane. And the first part we're going to talk protein. So the channel protein spans the entire length of the bilayer, and its role is to, well, as it suggests, offer a bit of a channel through the bilayer from the internal environment to the external, or the external to the internal, depending on the direction of flow. Now, we're going to talk about what these things channel, but we know that channeled proteins have the role of transport. And that is important because the proteins in our plasma membrane have several roles, and one of them is in fact transport. As well as these transport proteins, we also have another type of protein as well. This one is what we call a carrier protein. It's, gee whiz, my writing is not great. It also has the role of transport, but it does it in a different way. So carrier proteins will open and close depending on what they're exposed to. So if you can see, it's kind of like this would be the open to the internal and closed to the external, and then as we need the protein to do different things, so it works by, the carrier protein works by opening and closing, different to the channel protein, which is always open to the two environments. So at the moment, this one would be open to the internal environment and closed to the external environment. But when we go ahead and we need it to, it then switches up. So if it had contents inside of it, this would now be released, or it could be accepting contents from the external environment. Channel proteins work in a different way to carrier proteins, and we'll be talking about that when we talk about the different substances that move through it in a couple minutes. The next type of protein is a receptor protein. This one is an enzyme or hormone receptor protein. What would happen is a signal would be inserted into the top here, into the active site of the protein molecule, and when that signal is received, it would then activate another enzyme at the bottom here to go off and send a signal within the cell. And there are vice versa. There's also hitting the signal at the inside of the cell and releasing a molecule to the external environment. So these are your signal receptor role proteins. We also have anchoring proteins on the inside of our cell for one of the organelles that I didn't discuss in the organelles video, which was the cytoskeleton. So the cytoskeleton is one of the organelles inside of eukaryotic cells that offers a structural role to the cell. They're protein filaments that anchor around the plasma membrane and help to give it form and shape. So this one here would be a cytoskeletal anchoring protein, or I just call them anchoring proteins for short, just because they're the points where the skeleton can attach inside the plasma membrane. And so this one has a structural role inside the plasma membrane. And this is the first of the proteins that you can see that doesn't go all the way through the phospholipid bilayer. So this isn't an intrinsic protein. This is an extrinsic protein because it just sits on the cell membrane surface. All right, on to the last few. So we have glycolipids, glycoproteins, and cholesterols. So these guys all have various functions as well. And the first thing that you can see is that they either live inside the fatty acid layer. So this is your cholesterols and All right, so now we're up to glycolipids, glycoproteins, and cholesterols. And again, you can see here that they don't span the entire length of the plasma membrane. They don't need to. So inside the fatty acid bilayer bit, so in between the fatty acid tails, are cholesterols. Having a hard time remembering how things are spelt. And cholesterols give the membrane fluidity, give it the ability to bend and move around as much as they do, and allow there to be gaps in the membrane without it just being a complete mess and spilling out everywhere, spilling contents out everywhere. They also act as a bit of an anchoring point or a hold to get holdfast for the fatty acid tails, allowing the fatty acid tails to find a like substance to anchor to, because these guys are also non-polar by design. While on the surfaces, as you can see, pointing outwards, specifically going out into the external environment, are your glycolipids and glycoproteins. Glycolipids being glucose molecules or carbohydrates that are attached directly to the lipid molecules in the cell membrane. So they're either attached to the phosphates or to the actual lipid tails, and then the phosphate head would not be there. And glycolipids. Come on, pen. There we go. Glycolipids, attaching to the lipid molecules, and then we have our glycoproteins. These guys have the role of communication between different cells. So specifically in our immune system, letting white blood cells know that our cells are actually of the same type. So they're cell signaling molecules. They have the role of anchoring together, so they will anchor to other cells and say, hey, I'm like you, attached to me, we can exist together, and living in harmony. So these guys also have a really, really, really important role in cell recognition. Woohoo! All right. Putting it all together, and it looks something like this. It's a jigsaw puzzle. Well, not even a jigsaw puzzle, it's a mess. There's bits and pieces everywhere. There's cholesterols, proteins, glycoproteins, glucoses, all these things floating around everywhere. And this is one of the most important theories for cellular biology, and that is that our cell membranes actually exist as a model, and we call it the fluid Mosaic model. Not keeping up today, are you, pen? Fluid mosaic model. It's the theory that all of these bits and pieces, not randomly, they strew out throughout the membrane, allow the membrane to have some form of fluidity and movement to it, to give it that kind of characteristic that we can, in fact, have it bend and move and have different pieces all over the place. So this is, quite an important fact. Quite an important thing to kind of know about when we start talking about the plasma membrane. And often there are questions about what is the fluid mosaic model? Can you explain it? Or how does this fit into the fluid mosaic model? When people write tests and exams and things like that. All right. So that's the structure and the function of a lot of the bits and pieces of the plasma membrane. Now we're going to be talking about how do we get things to move across the plasma membrane. And, before I get to there, this is the kind of thing that I would get students to draw into their books to try and, talk about, the various parts, to talk about the fluid mosaic model, and therefore have this as a labeled diagram into their books. So it's, kind of, an important one. This is one that I sketched into my book, so it's not a student's piece of work, it's mine, but gotta model your own notes. All right, before we start talking about the types of membrane transport, I would get my students to fill in a document like this, like an A3 poster. I'd print them out double-sided so that they would have everything there, and we're going to be talking about various things throughout this entire process. As you can see, not only are we going to be talking about the methods, we're going to be talking about whether it requires energy or not. Give yourself a brief description of it, and then the types of molecules that actually use this type of transport. And we're going to get started with the simplest, of membrane transports. So here we have it. We have a number of molecules here. These guys floating around the outside are oxygen molecules. We can see that they are O2. There's two oxygens attached to each other, and the ones on the inside here are CO2. And I've taken a small snippet of the membrane and I've just placed it there so that we can show how these things are going to be moving across the plasma membrane as we need them. Interesting thing about oxygen is that it's very, very small. Same with carbon dioxide. They're both very, very, very small. And although they're polar substances, the interesting thing about oxygen is that it is very, very small. So is carbon dioxide. It's really, really, really small. Oxygen being smaller than carbon dioxide. The other thing to note about them is that they have no hydrogens attached to them. So there's no charge. They're completely chargeless. And so these guys have the amazing ability to just move through the plasma membrane. And they do this because oxygen and carbon dioxide, whoopsie, I erased that too early, so I'll put that back on there, O2 and CO2, exist in different concentrations on either side of the barrier, on either side of the cell membrane. This difference in concentrations is what we refer to as the concentration gradient. No, there's that maths word coming into it again. Doesn't happen often, but a colleague of mine made a joke that science is just applied mathematics. So here we go. So if you think about maths, you can think about gradient being the slope of our linear graphs, and therefore the difference from one point to another. Well, that's the same in this concept here. We have a difference in concentrations across the membrane. For example, here in the external environment, we have a high concentration, or relatively higher concentration of oxygen molecules than inside the cell. We only have that one lonely one in there, while we have the relative 3 on the outside. While the opposite can be said for carbon dioxide. There's a relatively high concentration inside of the cell, while outside of the cell, there's not much. And so this creates a concentration gradient. We have an area of higher concentration on the outside for oxygen compared to an area of lower concentration on the inside. So this is an important factor. Because of this higher concentration, oxygen is going to move with the concentration gradient. What that means is oxygen is going to want to move from where it is higher in concentration to where it is lower in concentration. To do this, it doesn't need any energy. There's no energy input. And because there's no energy input, we call this form of transport passive transport. Okay? Because there's no energy input, it is known as passive transport. The oxygen will move through the plasma membrane. Because it is small, because it's non-polar, it doesn't have any of those, it's not charged, it doesn't have any of those hydrogen molecules attached to it, creating different charges. It can easily bypass the fatty acid tails, which is the important part of moving through the plasma membrane. You need to be able to get through the fatty acid tail barrier, not so much move past the phospholipid head barrier. The phospholipid heads have lots of gaps, while there are smaller gaps through the fatty acid tails, and they always want to repel things that they don't like. This form of movement is very simple. My drawings are now going to go away so that we can watch the animation. So this form of movement's very simple, as you can see. Just move straight through the barrier. Move straight through with the concentration gradient. Those oxygen molecules go from the area of higher concentration to the area of lower concentration, and now all exist inside the cell. While the carbon dioxide molecules, they do the opposite. They'll move outside with their concentration gradient. They move with their concentration gradient. So where a higher concentration of carbon dioxide moves out of the cell in a similar manner. This form of diffusion is the simplest, and it's known as simple diffusion. Diffusion, come on now, diffusion being how we just move things across a membrane. Or actually, diffusion can be moving across anywhere. You can diffuse tea into water. It moves from an area of higher concentration to an area of lower concentration all the time. Okay, so simple diffusion, check. Let's move on, shall we? Oops, one more carbon dioxide. All right. So what happens when we have molecules that are not as simple? So here we have some glucose molecules. They're quite large, and they're large. They're 6 carbons. They're 12 hydrogens, and they're 6 oxygens. Now, although these guys probably don't have a charge, the way that they end up structured makes them polar. They actually have an area, much like water does, an area of positive charge and an area of negative charge. So that means that when they try to get through this barrier, they get repelled away by the fatty acid tails. The fatty acids don't want them moving through. Plus, they're too large, so you need a substantially large gap to open up. Similar for charged particles like ions. So here we've got our sulfur chloride ion. It's been separated from the sodium, like we talked about in our water lesson. Gets separated. The oxygens will be attracted to it. This guy will be relatively small, given any other situation, but because of all the waters that are attached to it, it actually makes him quite large. So again, he can't really move through the barrier. Also, being that he's a charged ion, if it was trying to get through the fatty acid tail, again, it would get repelled. Fatty acids really don't like charged molecules. They won't let them past. And so now we have this concentration gradient. We've got an area of higher concentration on the outside, an area of lower concentration of these molecules on the inside, and we don't really have anywhere for them to go. We don't have anything for them to do. And so, but we spoke about one of the proteins from earlier that would be able to assist us with this. And that's one of the channel proteins over here. Like, these are always open to the internal, external environment. They're kind of like they're a little bit like a one-way street, depending on what type of channel protein there are. But the majority of them are two-way. You can move in and out, in and out, in and out as needed through these channel proteins. And they act as a gateway for these molecules to move through. I've had to remove the water molecules because the animations wouldn't work properly. But here we are. We're moving through, moving through, and we moved against the with the concentration gradient again. And there's the glucose moving through. So I'll go back one. And so this here, again, there was no energy required, so it's still passive. We were using, the types of molecules were glucoses, large molecules, large polar molecules, and small charged ions such as chloride. And this one here used a channel protein. It was facilitated diffusion. I know you guys are probably saying this right a lot quicker, but yeah, I'm getting a little bit of lag going on with the computer. Anywho, facilitated diffusion is what we just watched there. And it still moved with the concentration gradient, had no energy input, and you just use the channel protein instead of using just going straight through the membrane. All right, so there are two forms of passive diffusion for molecules, for dissolved molecules, or molecules that exist in our membranes. Simple diffusion and facilitated diffusion. Next up, we're going to talk about what happens when we try to get molecules to move against the concentration gradient. So here we have a bunch of charged ions again. Let's call them chlorides again, and they could be any molecule really that needs to move against the concentration gradient, and therefore it has to go from an area of lower concentration to an area of higher concentration. And you can probably see here as a bit of a spoiler is that this is going to be the first of our transport methods that requires energy. I'm just going to click through the animations to get us through there. So, if we need to move something outside of the concentration gradient, well, we can use our carrier protein. So we've loaded up the carrier protein and then whack, we hit it with some energy. Energy in this case for cells is in the form of ATP, or adenosine triphosphate. Boom, hit it with some energy, and now the carrier protein does its thing. It'll open up to the external environment. The energy goes away, and the molecules are able to go out into the external environment, moving against the concentration gradient. Now, this is important for a lot of cellular processes. One thing that I can think of is actually the creation of ATP requires us to move molecules outside of the cell against the concentration gradient so that they can go through a channel protein to spin a wheel to combine to make more ATP. But anyway, I've got to spend a little energy to make a little energy. So this one requires energy. Yep, requires energy. That worked. So I wonder why that's working when I write like that. Anyway, I'll figure this out later. So it requires energy. It used a carrier protein to do it. Oh dear, I'm just going to put carrier pro like that. So it used the carrier protein to do it, and it moved against the concentration gradient. This one was active transport. Active transport is the general term that we gave to any type of movement that requires energy input to move molecules. We tend to think of it specifically as this version though, whenever we're talking about transport through or around the cell membrane. We tend to think about just active transport being this version, using a carrier protein to move molecules against the concentration gradient. Okay, nearly there. One of the last forms of transportation that we're going to talk about is the movement of molecules that are produced by the cell in bulk amounts. And because of that, we have to move a lot of molecule all at once. I'm going to introduce you guys to this one. This fella here is a vesicle. A vesicle that is a ball of essentially phospholipid bilayer, or phospholipids, that is going to come and merge with the phospholipid bilayer with the cell membrane. So this vesicle may have come from the smooth endoplasmic reticulum, may have come from the rough endoplasmic, probably came from the Golgi. Let's be honest here. The Golgi is the one that's responsible for exporting out of the cell. So it's going to be a group of proteins, hormones, whatever the cell has created. It's been packaged up by the Golgi and it's been sent to the plasma membrane. The Golgi packages it all up in a ball of plasma membrane. This is what we call bulk transport. The general term for this is bulk transport. And this particular one is going to merge with the phospholipid bilayer. And then all of the molecules will escape and head off into the external environment. This is what we call exocytosis. I'm going to write it down. So we have exocy, it's a Y, cytosis. Exocytosis is bulk transport to the external environment. So it's exiting the cell. EX for exit the cell, EX for exit, EX for exocytosis. The opposite is also true. We also have endocytosis. EN for endo, EN for enter the cell. Now, endocytosis isn't too much more complicated. It's basically exactly the same process, but in reverse. So, I could click through, all the molecules go in, and then it would close up, forming this vesicle, and the vesicle would then hightail it back out to the external environment. I'm going to click through my animations again just to kind of get through it. There you go, exocytosis again. All right, lucky last one to discuss. And it's probably a big one. It's a doozy. We're going to have another video on this particular one because there's a couple more concepts to get through. But ultimately, we've spoken about the movement of molecules. Now we're going to talk about the movement of water. How does water move across the plasma membrane? And there's a few things to take into account, a few things to think about, essentially, about water. And the first thing is that water makes up the majority of our bodies. There's no real difference in water concentration between our internal and external environments of our cell. The difference is when we have dissolved particles in our cell or outside our cells, that's when water starts to change its concentration as well. As you can see here, we've got we've got a few different versions of water going on here. So we've got our water molecules. The yellow dots I'll call sodium, and then the green ones are chloride. So we've got some dissolved salt inside of our cells, and we can see that the water molecules are attached to these dissolved ions inside of the cell. And because of that, they're essentially useless. They're not contributing to the water content of the cell. They're taken up. They're occupied. So, because these guys are occupied, and because they're currently attached to these water molecules, they don't offer to the concentration. Osmosis is what we're about to talk about. Osmosis is sorry, let me just write that out. Osmosis is the passive movement of water. So it's a passive movement of water. There's no energy required to move water. It moves against the solute concentration, but it moves with the water concentration. Okay, so water moves from an area of low solute concentration. So there's not much solute, there's not much molecules, particles in the outside of the cell at the moment. It's going to move from this area here through the membrane to an area of higher concentration. Now this is important. You really do need to specify which concentrations you're moving to. Essentially, inside of the cell there is a low water concentration because all these water molecules are occupied. But outside there's a high water concentration. So you move from an area of high water concentration to an area of low water concentration. But the molecules are moving from an area of low solute concentration or low particle concentration to an area of high solute. So that is against the solute concentration, but it's with the water concentration. I realize I've said the word concentration a lot in the last minute. Bear with me, please. All right. So how does this work? How's this process going to be achieved? Well, it's not as complicated as all that. Let me just turn off that. Thank you. Water, once it's established where it's got to move, simply moves through the plasma membrane, much like simple diffusion. There are a couple other theories though. There is another theory that there is a particular water channel protein called an aquaporin that allows the molecule to move through the plasma membrane, though the aquaporin is not part of the Unit 1 biology course. It's not something that we usually discuss, but just to let you know that if you do go looking out in the wild world of the internet, you may hear about the aquaporin, and it is a specialized protein molecule that allows, or a specialized area of the plasma membrane that allows water to move through it. But it is widely known that water, being a small molecule, can in fact slip through the plasma membrane and past the fatty acid tails. It just needs a relatively stable gap in the plasma membrane to do that too. I like to think that there is a molecule that it does slip through, because it seems to make sense in my mind, because fatty acids would repel water a lot, but fatty acids also don't like charged molecules. Either way, time for the lesson recap. I know it was a really big lesson. We're hitting over the 30-minute mark, so thank you for bearing with me. We discussed about the plasma membrane. We talked about the components and the importance of the phospholipid, the proteins, the glycoproteins and glycolipids. We went through membrane transport, simple diffusion, facilitated active endo and exocytosis, and osmosis. And I did not achieve my goal of doing this in a single take. So hold me to that one if you really do feel like it. Thank you, and I'll see you in the next one.